means biology. Today let's discuss about test cross. What is test cross? A cross between F1 progeny and the recessive parent is called test cross. A cross between F1 progeny back to its recessive parent is known as test cross. For example, let's take one pea plant with violet color flower and another pea plant with white color flower. If we cross between the violet color flower and the white color flower, here the violet color is the dominant and white color is recessive. Capital W stands for the violet, that is pure violet and dominant, and small w represents the white color and it is recessive. Capital W stands for pure violet, this is genotype of pure violet, and small w is the genotype of white, that is recessive. If we crossed between these two, in the F1 generation we found the violet color flower, that is in the heterozygous form, that is the genotype is capital W and small w. Okay. In the F1 generation we found violet color flower which is present in a heterozygous form. Okay. Then if we cross between the F1 progeny, that is capital W and small w, with its recessive parent, that is small w and small w, this is known as test cross. Okay. When we cross between the F1 progeny, that is the violet which is present in a heterozygous form, and the recessive parent is known as test cross. Okay. In case of monohybrid cross, the ratio of test cross is 1:1. Ratio of test cross for a monohybrid cross is 1:1, and for dihybrid cross it is 1:1:1:1. Okay. The ratio of test cross for a monohybrid cross is 1:1. But in case of dihybrid cross, the ratio will be 1:1:1:1. Okay. So, instead of self-crossing, why Mendel crossed the F1 generation or F1 progeny to its recessive parent? What is the reason of test cross? Why Mendel did test cross? Why Mendel devised the test cross process? Let's see. To determine the genotype of an organism showing dominant phenotype, Mendel devised test cross. To determine the genotype of an organism showing dominant phenotype, an organism which is showing dominant phenotype character or morphological appearance is dominant, when we observe a plant is showing a dominant phenotypic character, to determine its genotype, Mendel devised the test cross method. To determine whether the dominant phenotype is homozygous or heterozygous, to determine either the dominant character present in a homozygous condition or heterozygous conditions, Mendel devised test cross. Okay. For example, violet color, it is a dominant character and the genotype is capital W. Again, in the F1 generation, Mendel found again violet color which is present in a heterozygous form, that is capital W and small w. So the dominant character, that is violet color, can exist in two forms. One is homozygous form, that is pure violet, and another is heterozygous form, that is hybrid violet. Homozygous conditions contains two similar type of allele, capital W and capital W. Heterozygous form contains two different type of allele. One is capital W, another is small w. But both are showing the character violet. Okay. So, to determine the genotype, whether the genotype is present in a homozygous condition or in a heterozygous condition, Mendel devised test cross. Okay. गोटे प्लांट जदि गोटे डोमिनेंट कैरेक्टर एक्सप्रेस करूची इन F1 जनरेशन और F2 जनरेशन, F1 जनरेशन हो किंबा F2 जनरेशन हो, जेतबेले गोटे प्लांट डोमिनेंट कैरेक्टर शो करूची आमे बाहर बाह्य आकार देखिकी फिजिकल अपीयरेंस देखिकी जानी पारुचे जे एटा होची कोन गोटे वायलेट कलर फ्लावर. तो आमे फिनोटाइपिकली जानिले बा मॉर्फोलॉजिकली आमे ऑब्जर्व करले जे एटा होची गोटे वायलेट कलर फ्लावर. बट वी आर अननोन अबाउट इट्स जीनोटाइप. आमे तार जीनोटाइप जानिने. सो टू प्रेडिक्ट द जीनोटाइप, टू डिटरमाइन द जीनोटाइप, to identify the genotype, either it is present in a homozygous condition or heterozygous conditions, Mendel devised test cross. Let's see an example for better understanding. Here, violet color can exist in homozygous condition, that is capital W and capital W, and also in heterozygous condition, that is capital W and small w. Okay. So violet color can present in a two condition or two form. Okay. In a monohybrid cross, if we cross with the recessive parent, that is small w and small w, with the homozygous violet, because we have no idea about the genotype, we don't know either the dominant character present in a homozygous condition or heterozygous conditions. Okay. So, if we cross between the recessive parent with the homozygous violet, then we can get the gametes from capital W, capital W, capital W and capital W, and here from small w and small w. If we draw Punnett square, here capital W, small w, capital W, small w, capital W, small w, capital W, small w. Okay. Then all flowers are violet. Okay. Okay. Homozygous conditions. From the homozygous, that is pure violet, the gametes are produced, that is capital W and capital W, and from the recessive parent, the gametes are produced small w and small w. By drawing Punnett square, we can get the next generation. Okay, and we observe that all the flowers are violet. Capital W, small w, that is violet but hybrid violet. Capital W, small w, that is also violet, hybrid violet. Capital W, small w, violet. Capital W, small w, violet. So all flowers are violet. Okay. If the unknown organism, unknown genotype present in a homozygous condition, then all flowers will be in the form of dominant character, that is violet. Okay. If we cross the recessive parent, that is small w with the heterozygous vowlet, then the gamete from the capital W and small w will be capital W, small w, and from the small w, small w, small w, if we draw the Punnett square, we can get capital W, small w, capital W, small w, small w, small w, small w, small w. Okay. So this is vowlet, that is hybrid vowlet, and this is also vowlet, hybrid vowlet. This is recessive. All alleles are present in recessive form, that is small w, small w. This is white. This is also white. So we can get two flowers with violet color and two flowers are white. Okay. So the ratio will be 2:2. 2:2. It means 2 are capital W, small w, and small w, small w. This is violet. 2s are violet, that is hybrid violet, and 2s are white. Okay. 2:2 is equal to 1:1. Okay. So the ratio of test cross in a monohybrid cross is 1:1. Okay. When the dominant phenotype present in a heterozygous conditions, then the results of the progenies are half are dominant, half are recessive. Okay. In the ratio 1:1. Okay. If the dominant phenotype present in a homozygous conditions, then all flowers are violet. If we cross between the heterozygous violet, if the unknown genotype is heterozygous conditions or present in a heterozygous form, then when we cross the heterozygous form of violet with the recessive parent, we get the next generation half are dominant and half are recessive. Okay. In the ratio 1:1. Okay. So the ratio of test cross is 1:1, and if the genotype present in homozygous conditions, then all flowers are express the dominant characters, and if the unknown genotype present in a heterozygous conditions, then all the flowers or all the progenies expressed half dominant character and half recessive character in the 1:1 ratio. So this is all about the test cross method. In the next video, in the next class, we will discuss about the deviations of Mendelian principle, that is incomplete dominance. Till then, keep watching and keep learning. Don't forget to subscribe my channel, that is AL's Biology, for more video. Thank you.